Every organism begins life as a single cell. The cell divides repeatedly to produce a cluster of cells. This cluster of cells forms tissues and organs to perform the different activities of life. Cell division is essential for growth. There is constant wear and tear of cells during normal body functions. These worn out cells are replaced by new cells, which are produced by pre-existing cells themselves. This process is called cell division. It is a fundamental characteristic of life itself. After attaining maximum size, the cell begins to divide. Cell division is vital for the life on Earth generation after generation. It occurs in all life forms on Earth, from the simplest, the single-celled amoeba, to the most complex, the human race. In today's class, we are going to study about cell division, structure of chromosome, and cell cycle. So, in today's class, we are going to study about only the cell cycle. So, next class, we are going to study about the cell division. First, what are these chromosomes? The nucleus contains most of the cell's DNA, which is organized into discrete units called chromosomes. Each chromosome contains one long DNA molecule associated with many proteins. This complex of DNA and proteins is called chromatin. However, as a cell prepares to divide, the chromatin fibers coil and condense further, eventually becoming thick enough to be distinguished as separate chromosomes. These chromosomes then readily pick up certain dyes and get colored, hence name chromosomes. Chroma means color, soma means body. Chromosomes are highly coiled and condensed chromatin fibers. Highly coiled and condensed chromatin fibers are called as chromosomes. Chromosomes are the gene-carrying structure that consists of one very long DNA molecule and associated with a protein. It is found in the nucleus of a cell. Now, so it contains the DNA as well as the associated with the proteins. This DNA and this proteins is together, we can see called as a chromatin. This chromatin later give rise to a chromosomes. So this is the brief introduction of this chromosome. Next we can study about the chromatin. The chromatin material that constitute the fiber is formed of two substances. First one is DNA. It is called deoxyribonucleic acid. It is about 40%. Second one is histones. It is a particular type of proteins. It is about 60%. So this DNA and histones together we call as a chromatin. A single DNA molecule is very large and hence it is described as a macromolecule. DNA it is a larger molecule, so it is called as a macromolecule. It is composed of two complementary strands. It has two complementary strands wound around each other in a double helix. It is a double helix structure. Each single DNA strand is composed of repeating nucleotide, which are made up of three components: phosphate, sugar, arranged lengthwise, and a nitrogenous base. Nucleotide, nothing but it has made up of three components: phosphate, sugar, that is called as pentose sugar, and another one is a nitrogenous base. These three together we call as a nucleotide. Here, nitrogenous base is attached to the sugar inverse, which extends to join the complementary nitrogenous base from the other strand. Thus, the two strands together make a ladder-like arrangement with the nitrogenous bases forming the rungs of the ladder. Here, four types of nitrogenous bases we can see: adenine, guanine, cytosine and thymine. Adenine, guanine, cytosine and thymine. Here, adenine pairs with the thymine or thymine pairs with the adenine with the two hydrogen bonds. Between the adenine and thymine, there is two hydrogen bonds. Guanine pairs with the cytosine or cytosine pairs with the guanine with the three hydrogen bonds. Here, adenine pairs with the thymine with the two hydrogen bond. Guanine pairs with the cytosine with the three hydrogen bond. So, this is the DNA molecule. Now, you can see the structure of a DNA molecule. In the first diagram, you can see the two strands. One strand is moving from 5 end to 3 end. Another is 3 end to 5 end. And in between, we can see the nitrogenous bases. So, this is the DNA molecule, double helical structure. Clearly, you can observe this DNA molecule. So, here we can see the phosphate, pentose sugar and nitrogenous base. These three together we call as a nucleotides. Pentose sugar, because it has a 5 sugar molecule. So, it is a pentose sugar. In here we can see this nitrogenous base is always joined to the sugar molecule. Here phosphate and sugar, it become a backbone of the strand. They are present in the strand region. So, it is a backbone of the strand, that is a phosphate and sugar. Here phosphate and sugar molecules are connected to each other. This nitrogenous base is always connected to the sugar molecule, not for the phosphate molecule. Here thymine is joined with the adenine with the two hydrogen bond. At the same time, guanine pair with the cytosine with the three hydrogen bond. Here nitrogenous base is always pair with this, joined to the sugar molecule. So, here in the diagram we can see, so pentose sugar, in one strand it is a corrected position, in another strand is an inverted position, because one strand is moving from 5 prime to 3 prime and another is opposite direction 3 prime to 5 prime end. So, that is why this is the structure. So, this is the structure of the DNA molecule. Next, histone proteins. Histones are the proteins that help in the coiling and packing of DNA into structural units called nucleosomes. The DNA strand winds around a core of 8 histone proteins. It is called histone octamer. Each such complex is called a nucleosome. An individual nucleosome can be imagined like a football around which a long rope is wound with 1 or 2 loops. So, these are the histones proteins. The histones proteins, it helps for the coiling and packing of DNA. So, that part is called as a nucleosomes. A single human chromosome may have about 1,000,000 nucleosomes. Each human cell contains approximately 2 meters of DNA if stretched end to end. The DNA molecule is a macromolecule, so it needs to be condensed in order to fit inside the nucleus. The entire chromatin fiber is coiled and supercoiled, something like the coils and supercoils we see in a typical telephone cord eventually form chromosomes. In diagram you can observe histones, it is surrounded to the DNA, so that part is called as a nucleosome. This nucleosome coiled and supercoiled, it becomes chromatin fiber. This chromatin fiber later it gives rise to a chromosomes, and we can see it inside the nucleus. So this is structure of the chromatin fiber and DNA. Next, structure of chromosomes. Each chromosome in its condensed form as visible during the start of cell division consists of two chromatids joined at some point along the length. This point of attachment is called centromere or kinetochore. Here two chromatids they join together in a particular point. That point is called centromere. It appears as a small constricted region. Each chromosome's centromere is located at a particular site. The centromere also serves to attach the spindle fiber during cell division. As the spindle fiber contracts, the sister chromatids are separated at the centromere, and each is pulled away from the other towards the two poles of the dividing cell. So you will study detail in the cell division. After the completion of cell division, the chromatids decondense and revert to their very long and fine thread like chromatin fibers. There would be as many chromatin fibers inside the nucleus as the number of chromosomes that appear during cell division. So this chromosome later decondensed and it once again converted into a chromatin fiber. So during cell division, chromatin fiber condensed, it gives rise to a chromosome. After the cell division, this chromosome decondensed and it once again form a chromatin fiber. In the diagram, you can observe human chromosome. The two chromatids they join together. That point is called the centromere, or it is called as kinetochore. So this structure of the chromosome. Chromatid it has a 2 arms. Chromosome it has a 4 arms. Okay, chromatid it has a 2 arms. Chromosomes having the 4 arms.
The structure of chromosome. Chromosome consists of similar structures called chromatids. They are identical and are called sister chromatids. A typical chromosome has narrow zones called constrictions. There are two types of constrictions, namely primary constriction and secondary constriction. The primary constriction is made up of centromere and kinetochore. Both the chromatids are joined at centromere. This is essential for the movement of chromosomes at anaphase. If the centromere of the chromosomes is damaged, such chromosome fails to move at anaphase. The number of centromeres varies from chromosome to chromosome. The monocentric chromosome has one centromere and the polycentric chromosome has many centromeres. The centromere contains a complex system of fibers called kinetochore. Each centromere has 2 kinetochores linked with chromosomal arms. The kinetochore is made up of protein fibers and microtubules which assist in the formation of spindles during mitosis and meiosis. All constrictions other than primary are called secondary constrictions. In a given set of chromosomes, only 1 or 2 chromosomes have secondary constrictions. The nucleoli develop from secondary constrictions, and such secondary constrictions are called as nucleolar organizers.
Why we need the cells? First one, for the growth. Every organism, be it a plant or an animal, begins its life as a single cell. This cell divides repeatedly to form a cluster of cells which start shaping for different functions to form tissues and organs. Thus, cell division is essential for growth. So because of this cell undergo the cell division, so it increases our height and weight of the body, and it will help for the formation of the tissues as well as the organs. Next, for replacement, there is always a wear and tear of cells during the normal body functions. For example, 2 million red blood cells in our body are destroyed every second. These are replaced by new cells formed through a division of their parent cells in the bone marrow. In plants, the old and dried leaves fall off and new ones grow out. So, if the worn out or damaged cells are replaced by the new cells by the help of the cell division, the pre-existing cells, it undergo the cell division and give rise to a new cell, and that new cell occupies the place of the old or worn out cells. For repair, apart from normal wear and tear of the tissues in the body, there may be accidental injuries. One may get cuts in the skin or fractures in the bone. Repair of such injuries is again through cells which divide, cover up the gaps and join the broken ends. Yeah, if there is any wound is there, that wound will within a few days, that wound will we will recover from that wound. So because of this cell division, this cell occupies the newly formed cell occupies that wound region and help for the healing of our wound. So for the repair purpose, we require the cell. For reproduction, reproduction also takes place through the activity of the dividing cells. Amoeba or bacteria just divide to produce two similar independent cells by mitosis. In higher forms, as in humans or in the banyan tree, special cells in the reproductive organs undergo a special kind of division, that is called meiosis, to produce sperms and eggs. The sperms and eggs receive only half the number of chromosomes of their parent cells, that is, one chromosome from each pair. This reduction in chromosome number is very significant. So I will explain it in the later. So for the reproduction also, so we require the cell, and cell has to undergo the cell division. When the sperm and egg fuse each other, it forms a single cell, that is called as a zygote. The zygote, when undergo the continuous cell division, it give rise to a many number of cells. That cells give rise to a tissue as well as the organs, and from that new organism will develop. So for the reproduction, the cell is required, and cell has to undergo the cell division. What are the genes? Genes are specific sequences of nucleotide on a chromosome that encode particular proteins which express in the form of some particular feature of the body. So genes, it express the feature of the body, that is called as a genes. They are the units of heredity which are transferred from parents to offsprings and are responsible for some specific characteristics of the offsprings. So these genes, it transfer the body features from parents to the offsprings. So that is called as a genes, and it is a sequence of nucleotide. So it is nothing but it is present in the DNA. It is a sequence of the nucleotide is called as a genes. Next, cell cycle. The cell cycle or cell division cycle is the series of events that takes place in a cell leading to the duplication of its DNA and the subsequent division of the cell to produce two daughter cells. After the process of mitosis, the cells formed are relatively small. They have a large nucleus surrounded by the comparatively less cytoplasm. Thus, cell during the process of cell division undergo the interphase stage and regain the size of the mother cell. The sequence of events that occur from the end of one cell division to the start of second represents one cell cycle. So in the cell cycle, nothing but it is the duplication of DNA takes place because the DNA has to be transferred to their daughter cell. So that's why duplication of DNA takes place. So that period we call as a cell cycle. And in the cell cycle, the many changes takes place in the cell. The cell perform the many tasks in an organized, predictable series of events, which is called as a cell cycle. A cell must grow, synthesize materials like proteins, RNA, etc., and duplicate its DNA before physically splitting into two daughter cells. Before splitting into two daughter cells, it has to prepare itself for giving all the requirements for their daughter cell. The preparation for the cell division, so whatever takes place, whatever the preparations are takes place, it takes place in the cell cycle time. So that's why before undergoing the cell division, the cell undergo the cell cycle. After the cell cycle, it will undergo the cell division. A cell cycle consists of two phases. First one, a non-dividing phase called interphase, and second one, a dividing phase called the M phase, or it is called as simply mitosis. We can see the interphase as well as the M phase, or it is called as a mitosis. First, we can study interphase. The two daughter cells produced from a mother cell are relatively small, with a full-sized nucleus but relatively little cytoplasm. These cells are said to be in interphase. During this phase, they prepare for the next cell division and grow to the same size as their mother cell. Since no change in chromosome is visible externally during the interphase, formerly it was called the resting phase of the cell. But in reality, it is not true, as the cell is quite active during interphase in synthesizing more DNA. In the interphase, the cell prepares itself for the cell division. So, synthesis of DNA takes place in the interphase. That we call as a resting stage, but because we can't see any changes visibly, but inside the many changes will take place. So, but we cannot make out by seeing externally. In the interphase, there are three phases are there. First growth phase, next synthesis phase, next second growth phase. First growth phase, it is called as G1. In this phase, RNA and protein are synthesized. The volume of cytoplasm increases because of synthesis of RNA and proteins takes place. Mitochondria in animal cell as well as the chloroplast in plant cell, they have their own DNA. So, in late G1 phase, all cells must follow one of the two paths. They may either withdraw from the cell cycle and enter a resting phase, it is called R, or start preparing for the next division by entering the next synthesis phase. Your first growth phase, the RNA and protein synthesis takes place, so volume of cytoplasm increases. Especially in the mitochondria and in chloroplast, they have their own DNA. In their organelles also we can see this synthesis of RNA as well as the proteins. In this phase, the cell will undergo there are two choices are there for the cell. First one is cell may undergo resting phase, or it has to undergo for the next division, that is for the cell division. If it want to undergo the cell division, it has to prepare itself. So, if the cell want to undergo the cell division, so in that condition, the cell will move to the next phase, that is called synthesis phase. Otherwise, it is in the resting condition. Next, synthesis phase, or it is S. More DNA is synthesized, the chromosomes are duplicated. In the synthesis phase, DNA is synthesized and chromosomes are duplicated. Here also replication of DNA takes place. So that is the synthesis phase. It is called as a S phase. Next, second growth phase. It is called G2 phase. This is a shorter growth phase in which RNA and protein necessary for cell division continue to be synthesized. Here, the synthesis of RNA and proteins continues. Now, the cell is ready to undergo cell division and enter the dividing phase of the cell cycle. After the dividing phase gets completed, the newly formed daughter cells enter into interphase again, and in this way the cell cycle goes on and on. Here, after the completion of the second growth phase, the cell undergo the cell division. After the cell division, the daughter cell will form. This daughter cell once again undergo the interphase. So, it is a continuous cycle. After finishing interphase, it undergo the M phase. After finishing M phase, it once again undergo the interphase. In the diagram, you can observe the cell cycle G1, S and G2. Here G1 is a little bit longer phase. Next, after the G1, if the cell may undergo the resting or it may undergo the next stage. If it undergo the next phase, it will undergo the synthesis phase. Next, G2 phase. After completion of G2 phase, it undergo the M phase, that is mitosis. In the mitosis, we can see the prophase, metaphase, anaphase and telophase, and then we can see the cytokinesis. So this is the cell cycle. Next, formation of the new DNA, that is called replication. During the S phase of the interphase, each DNA molecule duplicates in readiness for their equitable distribution in the two daughter cells during mitosis. For replication, the DNA double helix open at one end, making the two strands free to which new strands begin to form. And the process continues in a sequence for the whole length of the DNA. Here, I already said synthesis phase, S phase. Here, the duplication of DNA takes place. For the duplication of DNA, the what DNA is there, the two strands, it will separate. The separation of two strands takes place at one end. When it separates, for that strand, the new one strand will join. So, at the end, in the DNA, we can see the one is the old strand, another is the new strand. So, one is the old strand, and is the new strand. So, for the old strand, the new strand will form. So, that is called as a replication of DNA. And this DNA then enters inside the daughter cell.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
In this class, we have studied about the structure of chromosome as well as the cell cycle. In the cell cycle, we can see the two phases, interphase as well as the YAM phases, and we have studied only the interphase. Next class, we are going to study about the YAM phase. Here, one interesting thing, cell production and the cell death. In the children, new cells are continuously being produced and they outnumber the dying cells. Here, the formation of cell is more when compared to dying of the cell. So that's why the smaller children will grow very fast. When in an adult, cell population stays constant. The number of new cells produced equals to the number of cells dying. So in the adult, how many numbers of new cells will form, the same number of cells will die. So there is no loss or there is no gain. The number of cell formed is equal to the number of cell dying. Next, when in old condition, the number of new cells produced runs short of those that are dying. In old people, the dying number of cells is more when compared to formation of new cells. Formation of new cells is less in number when compared to the dying of the cells. So this is the cell production and the cell death.